All right. Hello, everybody. Um, this worksheet that I'm going to be going over is the worksheet that you're going to use to follow in order to prep the material that we're going to be using, prep the tissue that we're going to be using to extract the protein that we are trying to purify. That protein is known as LDH, or the full name, lactate dehydrogenase. Now, lactate dehydrogenase is an enzyme that takes the molecule pyruvate and converts it to lactate. What it also does is it takes lactate and produces pyruvate, NADH. Therefore, you have a product of NAD+. So this is an enzyme that processes pyruvate, the end product from glycolysis, and utilizes NADH to make NAD+ and lactate. Now, for our purposes, whenever we're doing our catalytic assays, what we're going to be doing is we're going to be doing lactate and NAD+ and converting into pyruvate and NADH. The reason for that is this NADH right here is something that we can observe and monitor by measuring wavelength at 340 nanometers. So we're going to be doing, I guess, kind of the reverse enzyme or the reverse reaction, if you want to look at it in that way. Now, for this experiment's purposes, however, what we're most focused on is we are focused on, well, how do we actually get there? How do we extract this protein? And this enzyme, we're going to be extracting from beef heart. And so the first part of this is actually to begin processing that beef heart tissue or that beef heart. And the way that we're going to do that is I'm going to get about 100 grams of that tissue and add it to approximately 400 milliliters of the sodium phosphate buffer. This is our pH, or our buffer, with a pH of about 7.5. This is also known as your lysis buffer, your extraction buffer, and sometimes I'll refer to it as a wash buffer. Now, those three terms in this case are going to be used interchangeably, and quite often you'll see them used interchangeably. So lysis and extraction, you could kind of look at those as, well, we're trying to lyse open the cells, we're trying to extract this protein. Wash, on the other hand, might be a little bit confusing, but we're going to be using that term whenever we're talking about chromatography. We're going to have proteins bind to a chromatography column, and we're going to be washing away proteins that do not bind to that, hence wash buffer. Now, with that in mind, we will be blending this tissue for 5 minutes in a 400 milliliter sodium phosphate buffer. We're going to be blending 100 grams of it in that 400 milliliter buffer. Now, that blending process is basically just going to shear apart that tissue, make this a very viscous, viscous mixture. This is going to be very pulpy. If you've ever done any sort of I know whenever I was a kid, we recycled paper in one of my classes in elementary school. And so you rip the paper up and you get this really pulpy mixture. That's what this is going to look like, albeit red. Now, this is something that we are going to have to process, because when you have that really, really thick mixture, think about the practical aspects of it. Would this be something that's very difficult to pipette? Absolutely, it will. So we're not going to try to pipette it, but rather we're going to process it, or begin processing it, by using a large refrigerated centrifuge. And we're going to have approximately 40 milliliter fractions. So we'll have approximately 40 milliliters of this per group. And that 40 milliliters, what we're going to do is put it in this large refrigerated centrifuge and spin it at 500 times G for 5 minutes. So that's 500 times the force of gravity for 5 minutes. Now, essentially what this is going to do is it's going to take this, I'm drawing a microcent, or not a microcentrifuge tube, a large centrifuge tube, a 40 milliliter centrifuge tube. And basically it's going to start out where the contents look like this. It's very viscous, it's very runny, it's got a bunch of junk in there to say the least. And after 5 minutes, what's going to happen is the contents are going to pellet. And so you're going to see probably a big pellet down at the bottom, but still it's pretty viscous. This is a mixture that still has a bunch of junk in it, a bunch of insoluble stuff. So with that said, some things have settled down, but you have a supernatant. So you have a clear supernatant and a little bit of a pellet. At this point in time, you're going to save 1 milliliter of the supernatant, and you'll refer to this as your whole cell extract. You're going to save 1 milliliter of that for future use. Future use being at the end of the period, or not the end of it, but the second half of the lab period. Now, when you are done using that sample, or sorry, whenever you finish that 5-minute spin, you've taken the 1 milliliter off. Now we're going to ramp things up quite a bit. The centrifugations are going to be much faster and they're going to go for longer. And the reason for that is because what we really want to do is get good separation of all of the things that are in the centrifuge tube. So essentially what we're going to be doing for this part 2 down here is we're going to take this right here. So I'm going to draw this as our initially processed sample. And you're going to spin it at a much higher speed. You're going to spin it, but your rate is going to be different. You're going to be spinning it 15,000 times, or 15,000 RPM, 15,000 revolutions per minute. So it's going to go spin 15,000 times in 1 minute. So if you wanted to break that down, that's like 200 times per second. Well, 250 times per second. And what that's going to do is that's going to take that mixture and make a very robust pellet, but then a pretty clear and homogeneous mixture up top. This supernatant you're going to save. Okay, so you're going to save 1 milliliter of the supernatant, and you're going to label it the 15,000 RPM supernatant. Now, this is where you definitely have a solution that's quite a bit easier to work with, quite a bit easier to pipette up and down. It's easier to mix and everything like that. With that in mind, what you're going to begin doing is you're going to have something that's easier to work with, easier to process. In addition to that, this pellet, that has a bunch of insoluble junk, that has a bunch of membranes, that has a bunch of sugars, that has a bunch of stuff that does not go into our solution very well. And so we've cleaned up our sample quite a bit. So we have a supernatant that is clear-ish with a red tint to it, but also it's going to have mostly, or most of our protein hopefully is in that, because if it's in that clear supernatant, then it's much easier to work with that protein fraction. Now I'm going to clear the drawing and I'm going to navigate down in this worksheet. Okay, so we did that first centrifugation at 500 times G. We did a second centrifugation, 15,000 RPM for 15 minutes. So we've spun this sample a grand total of 20 minutes. Now, one of the things that is worth thinking about is, let's say hypothetically that you had two otherwise identical samples. If you have these two identical samples and you spin one for, and the speed doesn't even really matter. Let's say you spin it for 15,000 RPM. You spin them both at 15,000 RPM. You spin one of them for 5 minutes, and you spin one of them for 45 minutes. It's pretty reasonable to say that what they look like at the end of the 5 and 45 minutes respectively, it's going to be different. And it might not be different to the naked eye, or it might be different to the naked eye, but the longer duration is going to cause more things to pellet out. And so what that means is that if you spin for longer and longer and longer, more things are going to pellet. Now, with that in mind, we don't really have an infinite amount of time in order to spin these samples. So we are, when we're spinning, separating based on solubility. Well, if you think back to the protein purification worksheet, when you're doing your initial processing of your protein samples, or that protein purification worksheet where we were working on, with that software, our initial purification led us to separate based on, or separate with ammonium sulfate. Now, when you separate with ammonium sulfate, you're separating based on solubility. So to some degree, if you are spinning, you're separating on based on solubility. And if you are spinning with ammonium sulfate, you have kind of a two-pronged approach to separate based on solubility. Now, with that in mind, that's what we're going to kind of pivot to. We're going to, for part 3, add the salt ammonium sulfate. So NH42SO4. Now, this is a salt that has very kind of coarse crystals. They kind of look like sand crystals, really. What you need to do is you need to get a mortar and pestle, and you need to grind that up into a very, very fine powder. Now, what we're going to do whenever we're adding this ammonium sulfate is we are going to be adding approximately 0.242 grams per milliliter of that solution. Okay, so if you remember, we started with roughly 40 milliliters. Now we've separated, we've got this pellet, we've got this clear solution. In order to figure out the precise volume so that you know the precise mass to add, you're going to get a graduated cylinder, and you're going to transfer the supernatant into this graduated cylinder. That will allow you to say, okay, I have 25 milliliters. Now, when you've transferred that supernatant into the graduated cylinder, the the centrifuge tube that you're using that I just put a little plus sign by, all that it will have in it is that pellet, because remember, you transfer the supernatant to your GC, your graduated cylinder, not gas chromatography. So what you're going to do is you're going to clean out that centrifuge tube. So you're going to get that pellet and discard it. You do not care about the pellet at this point in time. Once you have cleaned that centrifuge tube, you're going to transfer the okay. You are going to transfer the protein, the soluble protein fraction, back into that centrifuge tube. You know that it's about 25 milliliters because you measured the amount in your graduated cylinder, and you're going to add 0.242 grams per milliliter. So 25 times 0.242 grams. Now, what that's going to give you is that's going to give you a 40% saturation solution. And this is known as a 40% cut. If you remember when we were working with the ammonium sulfate in the protein purification software, we started out with a small percentage, like 40%. Then we added 50% of ammonium sulfate, 55, 60, 65%. And what we were trying to do was we were trying to precipitate proteins without precipitating our enzyme. Or we were trying to precipitate our enzyme, but not precipitate other proteins. So what we are trying to do is basically precipitate some stuff to clean up this protein sample. Now, this is known as a 40% cut. And so what that means is once we have this ammonium sulfate in here, we're going to again spin for, you're going to balance the centrifuge. You're going to spin at 15,000 RPM for 15 minutes. Okay, so basically, once we get to these high speed spins, the speed's going to remain the same, the duration's going to remain the same. All right, now after we have done this centrifugation with the ammonium sulfate, now what we're going to get is a pellet, but that pellet at the bottom of the centrifuge tube, well, that's a bunch of ammonium sulfate and hopefully a bunch of proteins that are not our protein. All right, so that is going to bring us to a point where we have a grand total of, well, we've saved 2 samples thus far. After the end of that 40% cut, we're going to save 1 milliliter from that supernatant. After that, we're going to increase the ammonium sulfate. And the way that we're going to do that is, so let me go back. Here's our centrifuge tube. We've got our ammonium sulfate. This is our 40% cut. We're going to take 1 milliliter to save as our 40% cut. After we've removed that, then we're going to vortex. And really the purpose and the goal here is to solubilize or basically mix up that pellet, get it back into solution. So you'll have that clear solution. And at that point, we're going to add approximately 0.116 grams of ammonium sulfate per milliliter. We're going to then spin again for 15 minutes at 15,000 RPM. That is going to result in another sample that has a pellet, and this is our 65% cut. You're going to save 1 milliliter from that. And so what you will have at this point in time is a total of 4 samples. You'll have 1 milliliter of the whole cell extract, 1 milliliter from the 15,000 RPM, 1 milliliter from the 40% cut, 1 milliliter from the 65% cut. After you've done that, we're going to get to our very last point where basically our centrifuge tube looks a little bit like this. I've got a pellet, I've got a supernatant, I've saved the supernatant. Now what I'm going to do, or what you're going to do very carefully, is remove as much of that solution as possible, but leave behind 5 milliliters. And this is a very rough estimate. 5 milliliters of the buffer. And so you'll see the pellet, you'll see the small volume buffer, and what you're going to do is resuspend the pellet. And when you resuspend your pellet, the aftermath of this is going to be a sample that's very rich in ammonium sulfate, and you're going to save 1 milliliter of that. That is your 65% pellet. Okay. When you have saved your 65% pellet, take the volume that remains in this centrifuge tube to your IA. Your IA will then pool all of these samples into something known as dialysis tubing. And we are going to dialyze those samples. And we'll get into dialysis a little bit later on, but that's what we're going to end up with. So you will save a total of 5 fractions, and then you will have the remainder of your sample pooled in this dialysis tubing. Now, the very last part of this is you are going to be doing a Bradford assay with all of these samples. A Bradford assay so that you can figure out how much protein is in each one of these samples. Now, you're going to do that so that you can then, next week, use these protein samples for catalytic assays. Because what we want to do is we want to figure out how much protein is in each one of these samples. And what I've asked you to do is to take approximately, let me circle this, approximately 160 microliters of that 1 milliliter of your wholesale extract, 160 microliters of the 15K supernatant, and so on. So this should be, I mean, in the event that, well, look at this as an opportunity to practice the Bradford assay again. You can use the data that you collected last week in order to kind of process these absorbance values, or you can put together a new Bradford assay or do a new Bradford assay following the exact same kind of procedure and protocol that you did last week. At any rate, that's what this upcoming week's lab is all about, what's expected of you. And it's very important this week to preserve your samples, store them in a refrigerator, make sure that your samples are labeled. These are going to be used for weeks to come because we're going to use these to basically pinpoint which sample should we proceed with. Should we work with the 15K supernatant, the 40% cut, 65% cut, and so on? We're going to do that, and then we'll proceed and say, oh, okay, well, it looks like lactate dehydrogenase activity is highest in the wholesale extract or highest in the 15K supernatant. So be careful whenever you're doing this. The 1 milliliter fractions that you saved, do not discard any of them. You should save 5 fractions, 5 1 mil fractions. Review this worksheet to know which ones those are. But as a recap, the wholesale extract, which is after the 500 times G for 5 minutes, the 15K supernatant, the 40% cut, the 65% cut, and the 65% pellet. Your IA will separately hold on to the dialyzed sample. All right, everybody. I hope that was helpful, and I hope you have a good rest of the week. If you have any questions about this experiment, please let me know. If you have any questions about any preceding experiments, for instance, the Bradford assay, please let me know. All right, bye.